students, welcome to today's lesson on the main steps of scientific method. There are a number of steps of scientific method, but in this lesson we are going to focus on the 7 main steps of scientific method. To understand what is happening at each stage of the scientific method, let us take growth of tomato seed in tomatoes as example. The first step to scientific method is ask question or identify the problem. In this step, we have to identify the problem that we are going to study. Based on our example, we know that tomato seeds germinate when they are planted. But why don't tomato seeds grow inside tomatoes? The second step is do background research. Before we start trying to do the whole investigation ourselves, we should first check scientific magazines and the internet to see if anyone else has looked into the problem or into similar problem before us. Based on our background research, we find out that there are substances in plants that control growth or germination, what we call growth regulators. After we accomplished a background research, we have to construct hypotheses. First, we need to define what is hypothesis. Hypothesis is an educated guess that a biologist thinks the explanation of an observation. So, based on the example what we have taken, we can develop a hypothesis as there are chemicals in tomatoes that stop seeds from growing while they are still in tomatoes. This hypothesis is testable by experiment. We think that it is a chemical that is responsible. So, how do we prove that? To prove this, we could try covering some seeds with tomato juice and others with water. We can look if anyone gets germinate. Based on our hypothesis, we can formulate prediction. So what is prediction? Prediction is an educated guess as how biologist thinks his or her experiment will turn out. So then we can predict what type of results we can get after we accomplished our experiment. In this case, we can predict that seeds covered in tomato juice will not germinate, as well as seeds covered in water will germinate. Then we should design and carry out an experiment to test the hypothesis. In this case, we should develop and design scientific methods and procedures to test the hypothesis and hence find out solution to the problem or questions. So, in case of our examples, we can develop experimental procedures to perform the experiment. First, put several tomatoes in blender. Second, filter the blended material through some muslin. Third, collect the tomato seeds and wash them in distilled water. Fourth, place 20 tomato seed in petri dish on filter paper and cover them with tomato juice that obtained from filtering the tomatoes. Fifth, place 20 tomato seed in a petri dish on filter paper and gets covered by the same volume of distilled water. 6. Place them in a growth cabinet that will keep the temperature and lighting conditions constant. 7. Leave them for 4 days. 8. Check the number that have germinated in each condition. And 9. Repeat the experiment several times to enhance the reliability of the results. At the fifth stage, we should analyze results and draw conclusions. At this stage, experimental results should be analyzed to draw conclusion. In this case, tomato seeds that sown or placed in petri dish on filter paper and covered with tomato juice do not germinate, but tomato seeds that placed on filter paper in petri dish that gets covered with the same volume of water gets germinate. From this, we can conclude that it seems like something in the tomato juice is affecting the germination of the seeds. So it must be a chemical in the juice that inhibits germination of tomato seeds in tomato juice. The sixth step is accept or reject the hypothesis. If the result is similar with the hypothesis, our hypothesis gets accepted, and if not, our hypothesis gets rejected. So in this case, it seems as if the hypothesis is along the right lines. The tomato juice will only contain chemicals but not cells, and it does reduce the amount of germination. So we accept the hypothesis. Lastly, we have to report the results to concerned body and leave our research works to others for further work. That means we must now decide whether or not to report the results to other biologists. Someone else might decide to take the work further and try to isolate a particular chemical from the many in the juice to find exactly what is stopping them from growing inside tomato itself. Here is the diagrammatic representation of each steps of scientific method. As we discussed before, the first steps of scientific method is simply ask a question or identify the problem. Here we have to identify the problems that we are going to study. The second step is do background research. You see, before we start trying to do the whole investigation, we should first check scientific magazines and internet if anyone else has studied the problem or similar problem before us. The third step is simply construct hypothesis. We should scientifically guess or predict about what we observe as sources of problems or events in the environment. The fourth step, as you see, test with an experiment. We should develop and design scientific methods and procedures to test the hypothesis and hence to find solution to the problems or questions what we identified. And then we have to analyze results and draw conclusion. Experimental results should be analyzed to draw conclusion. Sixth step here is accepting or rejecting hypothesis. Means in this stage, if the result is similar with the hypothesis, our hypothesis gets accepted. If the hypothesis is false or partially true, it might be rejected. Then the last step, report results. So we have to report the results to concerned bodies and leave our works to others for further work.